The immune system. The major functions of the immune system involve destroying pathogens, detecting and killing abnormal cells such as cancerous cells, and removing dead cells from other debris from the body.
The body's first line of defense, the skin and the mucous membranes. Keratin is also resistant to most weak acids and bases and to bacterial enzymes and toxins. Intact mucosae provide similar mechanical barriers within the body. Besides serving as physical barriers, the skin and mucous membranes produce a variety of protective chemicals. Acid. The acidity of skin, vaginal, and stomach secretions, the acid mantle, inhibits bacterial growth. Enzymes. Lysozyme, found in saliva, respiratory mucus, and lacrimal fluid of the eye, destroys bacteria. Protein digesting enzymes in the stomach kill many different microorganisms. Mucin dissolved in water forms thick, sticky mucus that lines the digestive and respiratory passageways. This mucus traps many microorganisms. The mucin in watery saliva traps microorganisms and washes them out of the mouth into the stomach where they are digested. Defensins. Mucous membranes and skin secrete small amounts of broad-spectrum antimicrobial peptides called defensins. Defensin output increases dramatically in response to inflammation when surface barriers are breached. Using various mechanisms such as disruption of microbial membranes, defensins help to control bacterial and fungal colonization in the exposed areas. The respiratory tract mucosae also have structural modifications that counteract potential invaders.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibodies and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to the C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagocyl, releasing digestive enzymes such as lysozyme and protease in the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Natural killer cells, also known as NK cells, are a unique group of defensive cells throughout the body that police cells' actions. The cells act as one of the key soldiers in the fight against viral and cancer growth. To perform their duties, the killer cells first bind to its target, aims its weapon, and then delivers a lethal burst of toxins. These toxins produce holes in the target cells and creates events to destroy the cancer cell membrane. The DNA is then spread into many pieces, inducing it to undergo apoptosis. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. This system, the NK cells act upon, is similar to the killing methods cytotoxic T cells use. Through this process, inflammatory responses are enhanced due to the potent chemicals the NK cells release during its killing process. This ultimately makes the cell one of the body's nonspecific defenses. The inflammatory response is an important nonspecific response against tissue damage. It benefits the body by preventing the spread of damaging agents to nearby tissues, disposes of debris and pathogens, alerts adaptive immune system, and sets the stages for body repair. The inflammatory response starts when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelium cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillaries is referred to as margination. The inflammatory mediators released by injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelium cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils then undergo dramatic changes in shape and squeeze through the endothelial wall and into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotaxis substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria. The ultimate goal of this process is to clear the injured area of pathogens, dead tissue cells, and any other debris so that tissue repair can take place so healing of the damaged area can now follow.
Antimicrobial proteins, interferons. Some infected cells can secrete interferons to help protect cells that have not yet been infected. Interferons are cytokines that interfere with viral replication, they modulate inflammation, and they activate immune cells. The interferons diffuse to nearby cells, which they stimulate to synthesize proteins that interfere with viral replication in still healthy cells by blocking protein synthesis and degrading viral RNA. Interferons are not virus specific. They protect against other viruses as well. Complement. Complements complement or enhance other components of both innate and adaptive defense. They are a cascade of interdependent plasma proteins, each activating the other in turn until the final product is formed. When activated, these proteins can mark cells for phagocytosis, promote inflammation, and kill some bacteria all by themselves. The enemy can be identified by finding chemical markers that should not be there and by not finding them. Fever. Fever is a systemic response to invading organisms that causes an abnormally high body temperature. Fever increases the metabolic rate of tissue cells and speeds up their repair process. Fever is an adaptive response. When leukocytes and macrophages are exposed to foreign substances in the body, they release pyrogens. Pyrogens raise the body's temperature above the normal 98.6 degrees Fahrenheit.